Okay. You guys see in the Krebs cycle right here? Okay. What is that word? Malate. Okay. You can buy a supplement called citrulline malate. Citrulline comes from watermelons. Okay. You can get citrulline malate. The citrulline produces nitric oxide, which helps in some instances with blood flow. Okay, there's nitric oxide, or there's L15, which is a precursor of nitric oxide in a lot of pre-workouts. They used to make a pre-workout called NO Explode. They're great at naming, aren't they? Right? But so the idea is the citrulline malate. The citrulline promotes vasodilation, promotes blood flow. And then there's malate, which is actually a thing that's in the Krebs cycle. And they will argue that maybe having that malate is beneficial to aerobic metabolism, because if I can get that malate into the mitochondria, I can have more malate here in the Krebs cycle, and it will actually enhance aerobic metabolic function. Okay. Now, that is something that we would call, okay, biological plausibility. Okay, it's plausible. You can make an argument, right? This is going to go into a place and affect something that is plausible could have an effect on metabolism. Okay. So one of the ways as researchers that we look at things and try to decide cause and effect, and does this do that or something else, is if it's not biologically plausible, right? If we can't come up with anything from the aloe vera that would do something in the Krebs cycle or do something in the electron transport chain, then maybe we shouldn't say that it's going to affect metabolism in that way. I can tell you guys that despite the malate being in the citrulline malate, there's not a lot of evidence when you test it that it actually does anything here. Okay, but at least it's plausible that it could. Plausible that it could, right? And so that's one of the ways that I want you guys to try to learn and to be informed consumers of things. Okay, it's at least biologically plausible that we give you more creatine in your diet, that maybe you can have more creatine phosphate in your meals. It's biologically plausible that if we give you protein powder and supplement that, you've got more amino acids to help you make more, muscle, right? It would not be biologically plausible to say, I don't know, like just pick something, we're gonna drink, I don't know, cherry juice or something, and that's gonna do some other kind of thing. Put that in there that does that. Okay. Does that make sense? You guys kind of understand that? Okay. We did a study in the lab at Massachusetts. We did a thing on citrulline malate a couple of years ago. There was a capstone group that did a project on all of that in the fall as well. Okay. We're going to hopefully finish up talking about aerobic metabolism today, which will then get you guys to the place where you can actually do all of the worksheet stuff for next Monday. Okay. I will apologize for the time. We finally got all the tests entered yesterday. Several of you are clearly not paying very close attention because some grades were entered wildly wrong based upon things. I don't think anybody reached out. So we've got those are updated. So all the stuff in Canvas should be up to date. Your grade should be your grade. Okay, so just keep that in mind. We left off on Monday talking about the Krebs cycle. Okay, we left off talking about what I want you to know is that pyruvate gets converted into acetyl-CoA, 3 carbons to 2. Okay, as we lose a carbon, we make carbon dioxide up there at the top on the right, and we reduce NAD to NADH. Then we make citrate, and it goes through the Krebs cycle. We make a little bit of ATP here. We grab a whole bunch of electrons, and we make some carbon dioxide. That's the Krebs cycle. Okay. We need those electrons though. We're only making a little bit of ATP right here in one step, but all of those electrons that we have grabbed We're going to take those to the electron transport chain in a different part of the mitochondria, and we're going to use that to make a ton of ATP. Okay, a ton of ATP. Okay. So an introductory thing on the electron transport chain. It's on the inner membrane of the mitochondria. And I'll show you guys a picture of where this is happening. Okay, its location is really important. The Krebs cycle happens in a certain place, in the matrix. Okay, on the very, very inside. The electron transport chain is in that membrane that separates the two layers in the mitochondria. That inner membrane is important because we're going to move electrons around from one side to the other. We're going to create gradient, and that gradient is going to drive or power the enzyme that makes the ATP. It's going to power the ATP synthase enzyme. Okay? The primary thing that makes up the electron transport chain are these proteins called cytochromes. Okay? Cytochromes. Cytochromes are, I don't want to use that analogy, that's a bad analogy. I'm going to be careful about the analogies that I use. Anyway, cytochromes love electrons. Okay? They love electrons because they contain iron. Okay? Much like hemoglobin. Having those, they're very positively charged, and they want to grab those electrons. So what we're going to do is, it's sort of like a like the old-timey game of telephone. We're going to give electrons here on one end, pass it to your neighbor. We move some hydrogen, pass it to your neighbor. We move some more hydrogen. And if we need more people, we don't have enough cytochromes, and we would go pass to here, and then to here, and then to here. We pass the electrons down. Okay, and as they get transferred, we drive electrons, or we drive hydrogen. Okay, that those positive ions that want to follow the electrons out, okay, onto the other side of the membrane, and then it makes it all the way down here to the end, and here sits the ATP synthase enzyme. And we have this big gradient of hydrogen ions that we pumped because of the movement of the electrons. We get to ATP synthase, and all the hydrogen flows back in. It goes down its concentration gradient, and that, in essence, spins the ATP synthase enzyme, drives it, and we make ATP. Okay? And then all those electrons that are moving down, need some place to go. Okay? And where they go at the very end, and this is why lack of oxygen becomes so important, is that the electrons move down, they get to the end, and oxygen grabs them. And they bind to oxygen. That oxygen becomes a little bit negative, and then that oxygen grabs hydrogen from all of those hydrogens that we're pumping back across, and it makes water. Okay? And that balances out our equation. Okay? Without oxygen, here at the end, there's nowhere for the electrons to go, and so they get backed up on the cytochromes, and the whole process shuts down. Okay? The whole process shuts down. So cytochrome to oxygen. Grab the hydrogens that we've pumped back in. Now we've made water and ATP. Okay? Does that make sense, guys? Kind of, sort of, maybe. I'll show you all another picture. Okay? Now, what I have down here in red is, I believe what the book says, is that for electrons that have come off of NADH molecules, we make about 2.5 ATP, okay, per NAD molecule, or NADH molecule. With FAD, because they donate their electrons on a different cytochrome, so it can't move as far, you only get 1.5. Okay? Now, as I said the other day, you don't make half an ATP. What that means is that most of the time with NADH, we're making either 2 or 3. They kind of averages out to 2.5. Or with FAD, we're making 1 or 2. Okay? And it averages out right about here. These numbers have changed like 3 times since I sat where you guys sat as an undergraduate back in the dark ages. Okay? they'll probably change again as we get better at measuring things. Okay, here is the electron transport chain. Okay, here it is. All right, here's the inner compartment down here on the bottom. This is called the matrix. Okay, here's the inner membrane that has the cytochrome. So this is the electron transport chain. Then this is the outer compartment, and then the outer membrane on the top. Okay, just as I had set up down here, this is always fun to say, I'm in the matrix. Okay, here is the inner membrane. The second row is the outer membrane, and the kind of walkway here behind everybody is the outer compartment. So the Krebs cycle happens where I am right now, in the matrix. Okay. And so we are making all of these NADH and FADH molecules in the Krebs cycle here in the matrix. Those electrons, if I'm in NADH, I come and I donate my electrons to a cytochrome. Okay, then we pass them down and everything goes. If I was in FADH, rather than going on the first person, I would go to the second person. And they don't get passed twice fall. Okay. So we donated cytochrome 1 for NADH and cytochrome 2 for FADH, and we move these electrons down. As they move, right, the electrons go here, and so then when they leave the NADH, the electrons leave, those hydrogens also dissociate, and then we move those hydrogens from here in the matrix into the outer compartment, generate that big gradient. And then we come down here, here's the ATP synthase enzyme. It allows those hydrogen to pass back through, and that in essence powers that enzyme that puts the phosphate then back on ATP. And that's how we make all this energy. So right now, in every cell in your body, this process is happening right now. Pyruvate from glycolysis goes into the mitochondria, goes through the Krebs cycle. We grab all of those electrons, they move down, right? The electron transport chain and make ATP. Or we're going to take carbons from free fatty acid molecules and put those into the Krebs cycle. Do the exact same process. Grab electrons, pass them down the electron transport chain, make a bunch of ATP. Okay? But again, the Krebs cycle has many steps. The electron transport chain has many steps. So the process will make a bunch of energy slow. Process is slow. Okay? Okay. I like this picture because I feel like it wraps up and shows, at least from a carbohydrate perspective, what is happening in the big picture. Okay, you've got a cell, which is that kind of outer lighter yellow. You can see that we've got a glucose molecule in the cell. We're undergoing glycolysis, making a little bit of ATP there, right? We're then going to bring some electrons in from glycolysis. The pyruvate goes into the mitochondria, into the middle, into the matrix. We do the citric acid or the Krebs cycle. Right, we make CO2, we make a little bit more ATP, we get all those electrons down the electron transport chain, we make a bunch of ATP. Okay. So what you can see here is that for one molecule of glucose, through the electron transport chain, we're making 32 or 34 molecules of ATP. We've made a couple more in glycolysis directly, a couple more in the Krebs cycle directly. So you might make 36 or 38 ATP molecules by breaking down fully one molecule of glucose. Okay. So doing aerobic metabolism nets us somewhere in the low to mid 30s beyond what we get from glycolysis. If you make 2 or 3, we're going to make an additional 32 or 34 in the Krebs cycle and an electron transport chain, right? So that's 10 to 12 times as much energy doing it that way. So it's really, really beneficial for us to make it in the Krebs cycle. Okay, but it's slow. So you've got to balance then Am I making enough here? Can this go fast enough that I make enough here to meet the energy demand of whatever task I'm doing? And as you guys sit, absolutely that you can do that. For most of you, when you walk, absolutely you can do that. When you walk fast, you can probably do that. Okay? But then when you start jogging or you start jogging briskly, this is not enough. It's not fast enough for some people in some instances. And that's when we're going to have to start using more and more anaerobic metabolism and get over lactate threshold. Okay? Okay. So far we've talked almost completely, exclusively about carbohydrate. Okay? Glucose goes into glycolysis, and then it can make its way via pyruvate into aerobic metabolism. What this slide demonstrates, right, is some of the interactions between, okay, blood glucose, okay, glycolysis, which is going to be right here, fat stored in adipose tissue, and then amino acids that might be coming from protein. Okay, and so what you see here is 3 different substrates all going into basically into the Krebs cycle via this molecule called acetylcoenzyme. Okay, you can break down fat, you can get free fatty acids in your blood, take them into your cells, they go into the mitochondria, and you break them down 2 carbons at a time. Similarly, you can break down protein, you can make amino acids, you can put some of those directly, the amino acids directly into Krebs, or you can essentially convert the protein into glucose and get acetyl groups from there. And then you've got all the stuff that we've already talked about from a carbohydrate perspective. Okay, so the hub of all of this realistically is the Krebs cycle. We can only metabolize protein directly and fat directly in aerobic metabolism. Okay, only metabolize them aerobically. So that means during very high intensity exercise, during things where we need a lot of energy really quickly, these are not conducive substrates for providing us with ATP. Okay? For providing us with ATP. We will get into next week more about how we measure that balance between fat and carbs while you're exercising, and how we know that high intensity exercise uses almost exclusively carbohydrates in aerobic metabolism. And while you guys are sitting here right now, how many people eat breakfast? Okay, fair number of y'all. Those of you guys that haven't eaten breakfast are metabolizing more fat right now than the people that didn't eat breakfast, that did eat breakfast. Those of you that ate breakfast and say you went to Chick-fil-A or something, you got some hash browns and a chicken biscuit. One, you've now got your entire caloric needs for the day, which makes me very sad. Doesn't ever stop me from going, but you ate something that had more fat in it. You are metabolizing more fat because that's what you fed yourself. Okay, this is part of the idea behind a ketogenic diet or the Atkins diet or something. If you just eat fat and proteins and not carbs, you will break down fat and proteins. Okay, when you're in a fasted state, you metabolize more fat than you do carbs. Okay, we'll talk a little bit about that. We get the hormonal regulation of metabolism and things. It's due to hormone release, things like cortisol and glucagon, and some things that get released in between meals that help regulate blood glucose. Mostly you use the fat so you don't use your blood glucose because you haven't eaten any glucose. But that's kind of what all is happening. All of this goes through aerobic metabolism. Okay, it all goes through here. And so the reason that this becomes a problem in exercise is all of us have a month's worth of stored calories in adipose tissue for the most part in our body. Okay, some of us have more than a month. Okay, that doesn't do me any good because I can't get all that energy out of it during even one single long duration bout of exercise today. Okay, I will have to stop sometimes in exercise because I can't keep up metabolically, but it's due to a lack of carbohydrate. Even though I have all of this fat and all of this protein that I could use, it's the lack of carbohydrate that causes problems. So we'll get into a little bit of that food stuff as well. But I just want you guys to understand, this is kind of Krebs cycle is the hub. That's the middle place. This is basically the same graph from a different book. I know sometimes you guys like different things. I have tried to put in here where we're getting this 34 ATP from, try to count up where those things are coming from. Okay. All right. So what we're going to do now, we're going to talk a little bit about fat, a little bit about metabolizing fat. Okay. So depending on how much detail you want to go into, we're just going to skim the surface. We're just going to skim the surface. Sometimes we're going to call it lipid metabolism. You will sometimes see it referred to as a process called beta oxidation. Beta oxidation. Technically, beta oxidation is one step in lipid metabolism. You'll see it's one step. Okay. But it has gotten to be, because it's a really important step, and sometimes collectively people will say, are you doing breaking down fat via beta oxidation? That is kind of meant to encompass getting fat either from your blood or from adipose tissue into a cell, into the mitochondria, and then beginning to break it down in that beta oxidation step. Okay. To understand lipid metabolism, we need to go back to nutrition for just a minute and do a little bit more biochemistry. Sorry. Okay. Just a little bit more. You guys have heard of things called triglycerides before? You all cover that in intro to nutrition? Okay, good. The single most profitable class on campus, if anybody cares. Anybody wants to, I shouldn't say that on tape, right? With that, okay, triglycerides. Triglycerides are made up of what we call free fatty acids and a molecule of glycerol, right? Kind of looks like an octopus or a squid or something, right? You have that glycerol molecule that's kind of the base, and then you have the free fatty acids and these tails that shoot off on the back. Okay, it's the free fatty acids, right? They're going to give us all of the carbons that we're going to pull off two at a time to put into the Krebs cycle. You can put glycerol in there too, but it's really the free fatty acids. You tend to store lipid in adipose tissue as triglycerides. Okay, as triglycerides. So you have a little bit in your muscles, but mostly it's in adipose tissue. So my flabby tricep, okay, that's what we've got. Huge energy depot, right? 100, 200,000 kcals in some people more than that with all of this. Okay, other lipids are what we call phospholipids. These are neutral fats. This is the fats that make up the membranes, right, of your cells, okay? And then we have cholesterol, which is a whole other kind of thing that we could spend time talking about, but we're not going to. Okay, cholesterol. We are going to primarily talk about what happens with lipids, what happens with free fatty acids. In order for us to metabolize fat, you have to get a free fatty acid into a cell and into the mitochondria. We do that in one of two ways. Okay. The easiest way is eat something that has fat in it, digest it, and it goes into your blood, and then we pull it directly from the blood into a cell. Okay? You had Chick-fil-A this morning. An hour after you ate that chicken biscuit, then your triglycerides in your blood, the free fatty acid in your blood is going to rise. Some of that's going to get taken into the cells, and you'll use it for energy immediately. Okay. The other way to do this is to get it into the blood by breaking down adipose tissue that you have. Okay. We have to get the fat out of my flabby tricep, get it into my blood, so I can then put it into my muscles and use it. That breakdown process of getting it out of adipose tissue requires several steps, and it's pretty slow. Okay. It's pretty slow. We have to activate some enzymes that are going to break it down, okay? And those enzymes are activated by exercise. but it takes it a little while. During a bout of exercise, at about 30 to 45 minutes, we'll begin to see a rise in free fatty acid levels in your blood. Okay, it takes a little bit. So if you exercise for like 3 hours, you go walk for 3 hours. After about 45 minutes or so, the amount of energy you're deriving from fat begins to creep up slowly relative to carbohydrates, because it's taking that long to break down the adipose tissue and dump the free fatty acids into your blood. Okay? I'll say this here, but we'll come back to it a little bit later. It's difficult to say that cells have a preference for a metabolic substrate. But realistically, the preference of all of your cells that can break down fat is to use fat to get energy. Okay? We have a giant store of it. And we don't make any because it goes to the Krebs cycle. We do not make, there's no chance we make any hydrogen. We make any kind of negative metabolic byproduct when we metabolize fat. The other piece of that is the body does not regulate. Brain does not regulate or care about what your blood lipid levels are. It cares very much about blood glucose. If your blood glucose dips too far, you pass out and you go into a coma. Because your body is like, we're metabolizing too much, shut it down. Because the brain needs glucose. Okay? And so, if we can use fat, then we're not using glucose. If we can use fat, we're not using muscle glycogen. We're not using liver glycogen. We're protecting our carbohydrate stores, using our large store of fat that doesn't make any negative byproduct. Okay? So in that way, using fat is really, really beneficial. And that's why at rest, we use a lot of fat. Okay? This process here, if you look at it in your book, it's going to call it beta oxidation. What you see is a free fatty acid coming from the blood into a cell. We have to activate that fatty acid, which I think is the worst way to call it, but we have to do something to it to get it transported from inside the cell into the mitochondria. We make something called a fatty acyl coenzyme A, and then we move it and we take the acyl back off. Anyway, it's a stupid process, right? You put a thing on and then you move it and then you take it back off and you have exactly what you had before. It doesn't make a lot of sense to me biochemistry. It's like a wasted step, but that's how this works. Then you do the actual process of what is the technical beta oxidation step is. I have a free fatty acid. It has 14 or 16 or 18 carbons on it sitting inside mitochondria, and we beta oxidize and we pull 2 carbons off at a time. Clip 2 off, make an acetyl group, that goes into Krebs. Clip off 2 more carbons, that goes into Krebs. And you do that until you've clipped 2 carbons at a time and you've broken that entire free fatty acid molecule down. Okay? So if there's 14 carbons on a free fatty acid, let's just say, I'm going to go through Krebs 7 times to break that down. Okay? 7 times. So if I'm making, 30, 32 ATP every time I go through Krebs, and I gotta go through 7 times to break down 1 molecule, right? So I can do that now. It's a lot. It's a lot of ATP. Way more ATP to break down 1 molecule of a free fatty acid than from 1 molecule of glucose, because there's more carbons. Okay? And that's how this is going to work. Okay, that's how this is going to work. That's all that I want you to know. In previous classes, I've tried to teach all of those individual steps, and step 1 is this, and step 2 is this, and here's how we transport it in the blood. And I feel like it just it's too much. Okay, it's just a little bit too much. So that's all that I want you guys to know. We metabolize fat in the Krebs cycle by moving it from the blood into a cell, into the mitochondria, and then into Krebs. Okay. What I'm showing you here is a very famous relationship. I don't know that it's that important, to be honest. It happens all the time, but it was a big deal when people initially discovered it. What you see is on the On the y-axis is the percentage of VO2 max at which you're exercising. Okay, here's sitting on the couch. Here's walking very, very slowly, right? Jogging briskly, all out. Okay, 100% of VO2 max. And then the y-axis is going to show us if you add the blue and the kind of aqua dots together, that gives you the percentage of all of the ATP that you need at that particular intensity that's coming from carbs and what's coming from fat. Okay, so what you will see is that low intensity exercise or resting levels, we're getting 60–70% of our energy is coming from fat via aerobic metabolism, and then correspondingly a much lower, right, 25–30% coming from carbs. Okay, the vast majority of your ATP and aerobic metabolism at rest, as long as you're eating some fat, is going to come from fat. As exercise intensity increases, the amount of energy that you derive from fat and from carbs is going to shift. Okay, and this point in the middle here is called a crossover point. And so there is an exercise intensity that's going to vary among everybody based upon how well trained you are, how many mitochondria you have, what your diet has been, where we essentially reach 50–50 carbs and fat, and then as intensity increases, it switches, and we start using more and more carbohydrates. Okay? This is the effect of exercise intensity on fat versus carb breakdown. Okay? This has nothing to do with exercise duration. This is just in a vacuum, walking versus running fast. Okay, you're going to use more fat, or a greater percentage of the ATP will be derived from fat. Why do you think this is the case?
Fat will be metabolized fast enough to provide energy at higher reduced fat cells.
Good. Fat metabolism is slow. It's slower than carbohydrate metabolism. And so, as energy demand increases, as the percentage of VO2 max increases, we need the energy right now. And so, it takes longer to get it from fat. So, we preferentially then begin dumping more and more carbs into the system, because that will go faster. Okay? Because that will go faster. Now, what this looks like, if you overlay duration as at any given percentage of VO2 max, as you exercise longer and longer, then, right, the amount of fat that you use will get shifted up, okay? But in general, more fat at low intensity, less fat at high intensity. This led to a thing that I find to be hilarious, because the math doesn't work, okay? Anybody ever been in some sort of physique competition, or know people that compete in sort of physique competitions? Okay. What kind of cardio people do when they're trying to cut? Do fasted cardio, okay? But are they walking? Are they jogging? They're going hard. What are they doing? Long, slow distance. Okay. So there used to be, and I will joke, and I will say this very lovingly to everybody, because maybe some of you in here have done this, right? You've all seen the people walking up like the super crazy incline on the treadmill, holding on, like leaning back like this, right? Maybe some of you have done that. Okay, I at least get that we're making intensity go up. That makes more sense to me than the Stairmaster going sideways. I don't know what that does. Okay, what's the sideways Stairmaster? Are we working whatever these are, the abductors and the I don't know. Okay. The point is, right? It used to be a thing in the bodybuilding community that when you bulk up and then you get ready for your competition, you got to drop. You try to reduce fat, drop a little bit of weight, drop fat, keep your muscle, all of those things. That They would do cardio and they would walk really slow. Okay? For a long period of time. Why would they think if their goal was to lose fat, it should be to walk slowly? Why would you think that? Does that seem plausible to you guys? We'll tie it back to the other. Would walking slow, based upon this graph, maybe be a reasonably plausible thing to do if my goal was to burn fat? Yeah, you're right. Okay. Doing it fasted makes it even better. You guys ever try to work out in the morning, not having any breakfast? Feel like shit? Okay? There's a reason. You don't have any carbs, right? You're metabolizing the fat, and that is good, but it does not feel great. Okay? So you do this and you walk slowly, but it takes you 2 hours to go like 5 miles or something. You burn 100 calories a mile, give or take. Whether you're walking, whether you're running fast, whatever you're doing, it's about 100 calories. Okay? It varies a little bit based upon my miles I see. Okay? In that same hour or hour and a half, if you run, you wouldn't cover 5 miles, you'd cover 10. You'd burn twice as many calories. Okay? And if you do the math on this, even though at 50% of max, you're getting a little less percentage from fat, the amount of total calories you burn is so much more that you actually end up burning more fat than you do if you go really, really slowly and you do all of that. It's the total caloric sort of output that matters. And I'll be really, really honest, if you want to lose fat, it's really hard to exercise the fat away. Okay? I hate to break that to everybody. If you want to lose weight, you got to not go to Chick-fil-A. Right? Do what I'm telling you, not what I actually do. I'm gonna go to Chick-fil-A later. I got it. I'm gonna go to Chick. Okay. All right, Miles, you had a question. I answered it. Okay. I'm gonna finish here, okay, with a little bit of what we call rate control. This is why it's easy to illustrate that with fact. It's easy to talk about that with sort of what is going on with what happens with energy demand at low intensity versus high intensity. Okay. I call this the temporal or time organization or time control of metabolism. All metabolic pathways are active all the time. It's all about which one we get the most energy from at any moment. Okay. And so there is this push and pull of two things. The first is overall energy demand. Okay, overall demand. The second is speed of the pathway and the energy yield of that pathway. If demand is low, I can probably get enough energy from my slow pathways, the aerobic metabolic pathways that are slow, but they make a bunch. I can match that demand to what I'm making pretty easily. As demand increases, though, my aerobic pathways may be too slow. Or within my aerobic pathways, getting that energy from fat may be too slow, so it needs to come from carbs. It's all about finding that ability to match demand. Okay? We're only going to use anaerobic metabolism if we absolutely have to meet energy demands, because those stores are limited and things like that. Okay? Each metabolic pathway has a rate control step, a step or an enzyme or something that controls how fast it goes. Okay? And we need to know those so that we can understand the interactions of all of them together. Okay? Put all this up already, right? The speed of the pathways, creatine phosphate's the fastest. Why is it fastest? I heard it somewhere. Somebody said it. One step reaction. Okay? It's fast because there's one step. Glycolysis is the next fastest because it has the next least amount of steps. And then oxphos or sort of Krebs plus, right? Electron transport chain, aerobic pathways have way more steps, so they're even slower. Okay? Rate control of creatine phosphate is simple. It goes up when ATP goes up, and it's controlled by creatine kinase. Very simple. Okay? When you have ADP in a muscle, creatine phosphate will start to break itself down to match that. Okay? We tend to use this pathway once it gets going until it's gone, mostly because it's not going to last very long. Right? It's not going to last very long. Okay. With glycolysis, there are 4 key enzymes and steps that I want you guys to know. Okay? And glycolysis is more interesting, kind of like the aerobic side, because we can make it go faster or slower based upon different conditions. Okay? So, the 4 control enzymes in glycolysis are phosphorylase, phosphofructokinase, and then lactate dehydrogenase and pyruvate dehydrogenase. So we'll take these one at a time. Phosphorylase is the enzyme that breaks glycogen into glucose. It operates in the muscle, it operates in the liver. The interesting thing about phosphorylase and PFK is, sorry, more biochemistry, is they are what we call allosteric enzymes, in that if you bind certain things to them, you make the enzyme work faster or slower. Okay? And so we activate phosphorylase by putting it in the presence of calcium, by putting it in the presence of ADP, and by putting it in the presence of catecholamines, epinephrine and norepinephrine. So when you begin to exercise and you start turning over ATP, those ADP levels and then the calcium that's being released right in the muscle from the sarcoplasmic reticulum speed up phosphorylase and trigger the breakdown of muscle glycogen. Having the muscle glycogen broken down then dumps glucose 6-phosphate into glycolysis. So that drives demand on the top end of glycolysis. PFK is the slowest step. Okay, it's the slowest step. It is inhibited by creatine phosphate, hydrogen, citrate, and ATP. So PFK is at its slowest at rest. Okay, it's at its slowest at rest. We activate it using ADP, primarily inorganic phosphate. Okay, those 2 things. It's mostly ADP. So as you begin to exercise, as you turn over ATP into ADP, it makes PFK speed up. So now we're dumping more glucose 6-phosphate from glycogen into the pathway. We're speeding up PFK so we can move that glucose 6 through faster. Okay, which then means we can hopefully have more energy yield on the back. Okay, more energy yield on the back. Okay. Our other 2 are the dueling enzymes that control what happens to the pyruvate. Okay. We've made the pyruvate LDH, or lactate dehydrogenase, drives it into lactate, or lactic acid. Pyruvate dehydrogenase, PDH, drives it into the acetyl-CoA in the Krebs cycle. So the first 2 control how much pyruvate we're making, and then these 2 control where that pyruvate goes. Okay, where the pyruvate goes. LDH is not particularly allosteric. It comes in different isoforms or different flavors. So LDH in type 2 fibers works quicker than LDH in the heart or slow twitch fibers. PDH, though, is allosteric, and it can be sped up by the same kind of things, by ADP, calcium, and the like. Okay. So as you exercise, right, you increase glycogen breakdown, you increase glucose throughput in glycolysis, you speed glycolysis up, you also activate the enzyme that speeds that pyruvate getting put into Krebs. Okay. All right, we'll leave it there. I will see you guys next Monday. Okay? Enjoy the nicer weather. Make good decisions this weekend, and we'll see you next week. We are several people who are here soon. At this point, I don't even think we're gonna have our test next week. It's a little bit like on Monday, of what we have, okay?